Hello, everyone. Hope you had a good weekend. I can't tell you how my weekend was because I'm doing this on Friday. It's one disadvantage of doing this online thing is I got to do it ahead of time. Anyway, you'll notice the words on the screen say review, and we've made it to the review portion of our class. So the rest of the year we'll just be reviewing. The College Board, if you haven't heard, has decided to chop off the last 2 units of from the course. So we are not actually, we have been studying evolution the last week, but they have decided to chop that off. Although I have my doubts. They like it so much. Anyway, I picked this background because, well, going way back to Unit 1 in September. And you'll notice a lot of this we've repeated throughout the year. So hopefully this kind of, some of the stuff that maybe confused you earlier in the year will now make a little bit more sense. But this is, as you can tell, water. And I spent like 10 minutes finding a water picture, and that's what I got. All right, water. All right, so we have this oxygen, the hydrogen. It's a polar bonded molecule. It is essential for life on Earth, and has these three nice properties: cohesion, adhesion, surface tension, among others. We talked about others. And I want to highlight here the hydrogen bonding going on here. You have the unshared pair of electrons in this oxygen. You've got the polar charged hydrogen minus positive, opposites attract. And it's this hydrogen bonding that gives you a lot of the properties of water. Allows it to stick together. Allows an attractive molecules to attract at other surfaces. It allows the surface tension, the inward pull of the molecules with belly flopping. Should have gotten a picture of a belly flopper. That's water. Water is vital to life. Water gives us the properties. Without water, we're dead. All right, moving on. I got 15 minutes to finish this, and I'm going to move fast. All right, elements of life. It's kind of a lame photo, but really that's it. Carbon, oxygen, hydrogen, nitrogen. All right, carbon is used to build biological molecules, the backbone of living things. All those macromolecules are carbon-based. And we've got oxygen's primary component there. Nitrogen we haven't talked about a whole lot this year, but nitrogen is in proteins and nucleic acids. We'll see that in a minute. You also have phosphorus, which is not on there. Some lipids, like that phospholipid. Carbon, oxygen, hydrogen, nitrogen. All right, let's go to the macromolecules. Carbohydrates, the first one. What is the function of a carbohydrate? Anybody? It is a short-term energy. Remember that energy is stored in this carbon-hydrogen bond. That's your primary energy source. You get a little bit here, OHs. But that carbon-hydrogen, that's your energy. So carbohydrates. You have your base unit. Remember that monosaccharide. And you're going to have the bonds forming between them to form your polysaccharide or a starch, and it's gonna, we have our chain here, but yeah, that's called a glycosidic link there. Carbohydrates, these are isomers. I haven't counted them up, but I'm sure it's C6H12O6. So you have glucose here, and you have the energy being stored in these carbon-hydrogen bonds. And you can link them together to form starches. And remember, and this is amylose, but not, we were talking about cellulose earlier. Remember that link is important too for enzymes and digestion. You don't have to know any of the specific structures, but just know that they're there. We can break these bonds through hydrolysis, make them through that condensation reaction. All right, next one. I'm burning time. Lipids. Lipids. The function is long-term energy storage. A couple things again. Lipids are just a massive amount of carbon-hydrogen bonds, and that's how they're storing energy. You do have the difference between saturated and unsaturated. Right, it's unsaturated, don't have the maximum number of hydrogens, and you have this double bond in the middle, and they can have multiple double bonds, and that causes these not structures to be kind of branched or not linear, which leads to differences in state of matter at room temperature. Saturated can kind of stack together better, so they tend to be your solids at room temperature. Unsaturated tend not to stack, can't stack very well, and they are going to be liquid at room temperature. So your oils, or lipids, fats, oils, waxes. The other lipid I want to mention is the phospholipid. Talked a lot extensively about this year. It's got the phosphate, the hydrophilic head, and that's 3-, and then you have the polar tails, which is kind of the lipid portion. So you have a phosphate up here, the phospho part, then you have the lipid part down here. You got the 2 tails going off. This region at the bottom, hydrophobic. The circle part at the top is hydrophilic. Very important, as we learned in biological membranes, with that phospholipid bilayer allowing certain things in, out, while excluding others because, it can. I don't think I'm forgetting anything for lipids. Lipids are the one that are not a polymer. It's kind of an FYI. You do have. That's it. All right, moving on. Proteins. All right, proteins were a little bit more complicated. Remember, proteins kind of do everything in the cell. Without proteins, everything kind of breaks down. And I really want to go off on a tangent about bacterial toxins right now and how they can affect proteins, some of them, but I won't. Look it up on your own time. But proteins, your base unit for the protein, the monomer, is the amino acid. You got your N group here. You got your carboxylic acid here. You have the N-terminus and the C-terminus are your 2 ends to a protein, and you have this R, right? Because you can have any amino acid R group. Remember, there's polar, nonpolar, and charged. So those R groups can change. Those R groups really determine the structure. And changing it with the DNA, remember, if you change the DNA, you change the RNA, and then ultimately you can change what this R is, which then would conceivably change the protein, which then could lead to a new phenotype, which then could lead to natural selection. So a lot of it goes back to proteins and DNA, what we talked about. So a couple things with the, you get a peptide bond formed in here, and we have the carboxylic end and the nitrogen end form. So you get the carbon-nitrogen bond. They're going to release water. All right, you get an oxygen, you get a hydrogen, you get the OH, the hydrogen. You end up with that. But then there's the different levels of protein structure. Protein, or the primary structure, is just the amino acid sequence, how they're joined together, and just what the sequence is. The secondary structure are the alpha helixes and the beta pleated sheets, and this is due to some hydrogen bonding between the backbone of the amino acid. You got these oxygens and these nitrogens. So you got a little bit of a polar bond here. You got an unshared pair of electrons and nitrogen. So between them, they're going to be able to form some hydrogen bonding if they're wrapping around in different ways. Anyway, there's hydrogen bonding there. The third level is the tertiary structure, and this is due to the interactions with the R groups. Remember, the nonpolar R groups tend to orientate themselves into the core of the protein. The polar uncharged R groups orientate themselves out toward the outside, towards the water portion. But it's the interaction of the R groups that determine this tertiary structure. And then the quaternary structure is when you have multiple proteins kind of joining together. I saw something online that might be helpful. These are like letters, and the secondary is kind of like your words, putting letters to words. And the tertiary structure is like sentences, and the quaternary is paragraph. I don't know if that helps at all, but all right, I've got 6 minutes for the next macromolecule. Nucleic acids, our last one. Function. Here, remember, it's going to be our genetic carrier, the boss of the cell, so to speak. It's going to hold the information, key. All right, there are 2 types of nucleic acids, DNA and RNA. The nucleotide is made up of the phosphate group, that 3- charge. You got a sugar, a ribose sugar, and a nitrogenous base. And they're going to orientate themselves into this structure. And right here, I want to look at DNA first. DNA, remember, has the base pairing of ATGC. We have the pyrimidines and the purines. We have the, remember, the cut right here. These guys are the single ring, and then AG are the double rings. And the orientation and how they bond together. I remember with the 5 to 3 from DNA replication. But here we have a double helix, and a couple things I want to point out. Firstly, the nitrogenous bases, what holds the double helix together is the hydrogen bonding between these bases. And they are exactly right in order to bond. So, you know, it's always a single with a double, a purine always going with a pyrimidine. And the way that they are orientated match up perfectly, whereas any other combination is going to give you something that's not stable. And then as you look at the DNA molecule, this double helix, it is anti-parallel. So I have a 5. The 5 carbon is exposed up here as the end, and if you trace it down, the 3 carbon is on the other end. And then the strand opposite to it runs the opposite way. It's kind of anti. So they're kind of same direction, but anti. Anti-parallel. So you have the 3 at the top and the 5 and the other strand, and the bottom is going to reverse 3 and 5. But the base pairs are always complementary. So I think purple is C, and that's G. All right, so it's going to be complementary. Oops. I think that actually is all I wanted to say about nucleic acids. But now we can look at RNA. The differences between DNA and RNA. So RNA, remember, is single-stranded, and it has uracil instead of thymine. And it's not shown here, but also the sugar is different. DNA is deoxyribose, and RNA is just ribose, that ribonucleic acid. RNA is usually single-stranded. Bonus points, you remember where it can be double-stranded. It'll be a virus. And I think that's all. We got 12 minutes here. That's Unit 1 in 12 minutes. So everything you needed to know. It took us, 3 weeks to get through that information, and we can have done it in 12 minutes. All right. Let me know if you have any questions. Enjoy your day.